Yes, so today my topic is new breeding technology for targeted crop improvement. So I will talk about the current regulatory and market trends in gene editing technology with emphasis on our current projects, what we are working on, and what we could deliver to the community in the near future. So the gene editing technology has been awarded the Nobel Prize last year. The committee says that the technology is genetic scissors and can be used as a tool to rewrite the code of life. So as we know, the mutation is the key to crop breeding, and conventional breeding that takes about more than 10 years to introduce a mutation into a new variety. While the gene editing technology can induce the mutations in the targeted gene in the current varieties, and thus can reduce the breeding time. So based on the whole genome sequencing and gene identification, now we can modify any genes within the genome. And this technology has been successfully applied to all the major crops, and the new varieties with improved traits are being developed, such as the new wheat with pondering medial resistance, maize with drought tolerance, and the corona with herbicide resistance. Very importantly, the gene-edited products can be non-GM in Australia and many other international markets. So how does gene editing working? So suppose this is a target gene, and this is the nucleus. The nucleus is a genetic scissor. It can cut the DNA into two. Then the living cell will have a sense, gosh, my DNA is cut and I'm dying. No, I have to survive. I have to repair the break. So if this repair is spontaneous without any disturb, we call this is SDN1 mutation. So SDN1 is usually error prone. It will usually add more or miss some information at the breaking point, which means that it will usually end up with a different copy of the gene, and thus the mutation was created. Of course, we can add some template to guide this repair. And either it's a small template or a big template, because this is SDN2 or SDN3. In Australia, the organisms modified using SDN1 are not GMOs. This is already effective 2 years ago. So why gene editing will be accurate? If you look at here, this nucleus or the scissor is driven by a guide here. The guide is just function as a unique postal address onto a letter or on a parcel, so it can be delivered correctly. So now we can just set, change, or modify the postal address, and the scissor will be delivered anywhere as you wish. That's what I said, we can modify any genes within the genome. So to be non-GM in Australia, not only we have to use the SDN1 mutation, we also have to get rid of the foreign genes. If I still use the letter as an example, we only have the letter pages, but we have to discard the envelope or the parcel package with the address information here. So this is the global regulatory environment for gene-edited products. The green colors here just showing the countries having certain products excluded from GMO regulation, including United States, Brazil, Argentina, Australia, Japan, and the UK. And the yellow colors here just showing the countries they are imposing GMO regulation and ongoing review of gene-edited products. And we also have some blue countries such as New Zealand and South Africa. They have the policies that the edited products are under GMO regulation. So please be aware, we only have some certain types of the edited products excluded from regulation. In Australia, it's SDN1. In Japan, both SDN1 and SDN2 will be excluded. So what are the real products on the market? Please be aware there, here. For all the products are listed here, they are non-GMO in the corresponding countries. So the high oleic soybean oil was on commercial sale in United States 2 years ago, and most recently, just within 2 months, the gene-edited tomato was on commercial sale in Japan. This tomato has much high GABA content. So GABA is amino acid and has beneficial for human health. Just within 1 or 2 fruits daily, it can reduce the blood pressure. Okayama University has developed a new wheat with pre-harvest sprouting resistance. It's not commercial yet, but under field test, I believe it will be available soon. So this is the pipeline, how to conduct the gene editing. I'm not touching the details today. It is tedious and time consuming, and it usually takes at least 6 months to 2 years to have the mutants generated. But in the color boxes here, I just list all the key resources required for gene editing. And one important one is the reference genome. So the reference genome is our address book or the Google map. Without address information, the delivery goes nowhere. So the DPIRD is a key player for the barley, oats, and the lupin genome sequencing. We have comprehensive genomic information of WA's major crops. The major challenge here is how to induce the mutants from the current varieties. A common issue in cereal crops such as wheat, barley, and rice, there are very few varieties can be modified. I should say handful of varieties can be modified. And the majority of the commercial varieties, they are resistant to this process. As you can see here, this is the barley reference variety, Golden Promise. This is a Scottish variety, has been released 60 years ago. And in contrast, this is our commercial variety, La Trobe. If you compare with the reference variety, the size is much smaller and much fewer green plants could be induced here. So firstly, I work with my colleagues from Cyril Harper lab, and we established a new gene editing platform for commercial barleys. Now we can do SDN1 type mutations in both commercial and reference varieties at a high mutation rate, over 50%. And the majority of the mutations generated is just small mutations, pretty similar as the previous EMS mutation or gamma radiation mutation. So what do we have now? We have developed a small mutant library, and I call this is a treasure chest. So the pictures I put here is the individuals only with dominant morphological changes based on our visual phenotyping and observation, such as massive tillers and massive spike numbers with extremely high yield potential, and ultra long root with potential drought tolerance, broad leaf, early maturity, large green size, and dense spike type. I believe some mutants will also carry some invisible traits, such as disease resistance or stress tolerance to drought, frost, salinity, but it needs further screening. Waterlogging. This year in some areas, they have high rainfall and got waterlogging issue. The barley varieties do have variations in waterlogging tolerance. As you can see here, the germplasms on the right are tolerant, while the left ones are susceptible. So previously, my colleague Sue Bronton has mapped the tolerance QTL onto chromosome 4H, and now we have fine mapped this region into a very small region. If I still use address as example, previously we locate a city, but now we are zoom in, zoom in, zoom in. We have a suburb name and we have a street name. We only have a few properties there. So I believe in the future we're going to find the responsible gene, and we can use the gene editing technology to improve the waterlogging tolerance. I would like to talk a little bit more about our work in nitrogen use efficiency improvement in barley. So from agronomic data, we know that about 40% of the total soil nitrogen credit is just lost into environment. These 40% are not utilized by plants at all. From our large-scale screening, you can see here, there's a huge variation in terms of NUE among barley germplasms, from the lowest in yellow and the highest in the red. And the black arrows here just pointed out our most successful barley varieties, including Handmash, La Trobe, Flinders, Bullock, and Compass. You may notice that most of the varieties just fit into the low to medium nitrogen window. So one critical information I would like to highlight here is our current commercial varieties have at least 20% NUE potential to improve. So what is the genetic control of NUE in barley? We just fine mapped one gene. This is responsible for many traits under no nitrogen conditions. It's related to the shoot length, number of leaves, and biomass. Actually, this gene is a negative factor, which means that if this gene is functional, the NUE is likely to be low. But if this gene has been chopped out or knocked out, NUE may increase. So we have generated the targeted mutations within this gene. As you can see, there are few base pair deletions or base substitutions here. If we use this as original address, now we have this land has been subdivided or rezoned. Now we have 1A, 1B, not only one. So the mutants displayed very interesting and promising phenotypes here. As you can see, they are massive. They have a delayed leaf senescence. So from the detailed phenotyping, we know that they have a much high chlorophyll content in the leaves. They have high tiller number. they have high soil and grain weight and end up with a high yield per plant. So this phenotyping has been done at normal nitrogen levels. And limited nitrogen levels, they also have much high chlorophyll content. As you can see here, the mutants have a dark green color, while the original variety, it has a yellowish due to the nitrogen deficiency. One very important question, is this gene transgene free, or we have discarded the envelope or the parcel box? Yes, the answer is yes. We use markers to screening all the mutants here. As you can see, only very few mutants carry the transgenes, but most of the lines, they are transgene free. Now we are back crossing the advanced mutants with current commercial varieties, and in the late future, we're going to have more materials for wild field test. This is another piece of work we have done in the green revolution gene. And we have modified the green revolution gene in barley. As you can see here, they're massive. The spike number increased and the plant height decreased. They have a late maturity with an increased yield potential. So other than barley, we also have established wheat gene editing platform. As you can see here, this is a proof of concept study. We have a putative mutant here. This is much shorter and evergreen compared to the wild type on backwards. We are also adopting this technology for legume crops. We are developing new lupins with resistance to multiple herbicides. And we are also developing non-flowering leucaena to reduce its weed risk in the northern regions of WI, so as to promote this pasture crop for beef industry. To sum up, the gene editing technology is highly effective and efficient for crop improvement. And at DPIRD, we have established the gene editing platforms for WA's major crops. And we have advanced mutants developed, and they have the potential to be transformed into future varieties with greater profitability. So many thanks to the collaborators and contributors to the project. Thank you.
Yeah, for an old fart like me, I actually understood that, so you've done very well.
Thank you.
Any questions from the floor?
Oh, I understand.
Okay, no, because I understood it, I've got a question.
Yep.
So there are other ways to do mutations, chemical mutations as well. So is the gene editing that you're doing more efficient than doing it that way?
If you're talking about the mutation type, the gene editing is a pretty broad methodology. We can use SDN1 or 2 or 3. Like 2 or 3, they can do gene replacement for the common EMS or gamma radiation. They couldn't do gene replacement. But for the SDN1, they just generate the small indels pretty similar as the previous methodologies.
Okay, well, thanks for that. Still got time for a question if anyone wants to ask something? Okay, if you think of something, put it in Slido and we'll come back to it at the end of the session for all of the speakers. So thanks. Yep. I think you thanked him once, but let's thank him again.